genotype screening. I'll introduce that term of phenomics here. And it's another omics, but it really deals with whole organism interactions or reactions to their environment. Okay, so if we think about biogeochemical cycling in plankton, and we don't know a lot about that intra-specific variation, we can take a step back and look at the ecological effects of intra-specific variation instead of biogeochemistry. And I want to point to a really nice review paper that DeRoche et al. Published in 2017, looking at the relative importance of species effects versus intra-specific effects. And this is a review paper of many studies. Most of them are terrestrial, some are freshwater. And each of these dots here represents a different study. Points on this portion of the graph are studies that showed larger species effects. And surprisingly, points on this side of the graph are studies that showed larger intra-specific effects. So the effects of switching out genotypes in an experiment were larger than the effects of switching out species. And that's something that we don't think about a lot. And I asked my student, Stephanie Anderson, to pull up some of her data, and she sent me this as a sort of an example from a marine organism. This is 2 species of Skeletonema, and this is their nitrogen density, so in the micromoles per cubic micron. And what you see is that the intraspecific variation in this particular characteristic is much larger than the interspecific variation. So depending on what genotype we might be looking at, we might see larger intraspecific variation or effects than interspecies effects. So one of the things that motivates my research is really wanting to understand what that intraspecific variation looks like and how that plays out in an evolutionary context. So we can ask, what's going on out in the field? What's the evolutionary potential of these different organisms? As Elisa said, evolution will happen, right? What we want to understand are some of the key components of that. So what's the potential for that evolution? For example, what are the mechanisms? Is it drift, selection, sex? What are the rates of change? How does water circulation influence these processes, right? Because we're talking about planktonic organisms that are subjected to movement by tides and currents. And kind of to sum that up into one question is, what does that planktonic seascape look like? So I'm going to boil that down into 2 themes for today. The first is ocean forensics, where we're going to look at how much raw material is out there for evolution to act on, and how is it subdivided. This allows us to, we'll look at the temporal and spatial patterns of genetic diversity, and this will tell us about evolutionary potential. And the second component is to look at diatoms and their friends. And I put friends up there before I knew that Jeff was going to talk about all of these friendly interactions, so that's a nice follow-on. You know, one of my students said, you know, you can tell a lot by a diatom by who it associates with. I'm going to tell you a little story about that. That this happens not only in test tubes, but we'll be looking at the potential for microbial coevolution between diatoms and their associated bacteria. So what I'm going to do with this ocean forensics section is give you an example that is coming out of my lab to give you context, to tell you a little bit about the methods, and then we'll go out into the literature and look at what we see in other species and other studies to get a more holistic picture of what we think is going on. So the first thing I'm going to tell you about is a study that we did focusing on the diatom Thalassia xyrotula. Here's a nice picture of it. It's a chain-forming diatom, cosmopolitan. And that study was done by my former student, Carrie Whitaker, who's now here a faculty member at SEA. So you may see her walking around in Woods Hole. When we started this study, we wanted to see what kind of intraspecific variation was out there in this organism. And here's a thermal response curve, and you can see that there's quite some variation, both in terms of the thermal optimum as well as some of the edges of that thermal curve. So we knew there was variation. We wanted to see how that played out on a global scale. And so if we want to look at the potential for evolution, we want to understand the genetic variation that's out there in the field. And there are a couple different ways to measure it. One of those ways is to look at microsatellite markers. These are repeat regions in the genome. Here's a GT repeat. And those repeat regions vary in length. and that's a sure sign of mutation. I'm going to highlight when some of those evolutionary processes come up, right? Skill building here. All right, so we use many regions, we look at many regions of microsatellite markers, and we can then identify individuals. And that tells us how much variation is out there. We can do this in other ways too. We can do whole genome scans to look at variation across the whole genome. Here you see about 20 million bases of the Thalassius aerochula genome that we're looking at. I'm not going to talk about that today, but I can talk about that afterwards with people who are interested. We can look at many of these markers from many individuals in any one bucket of water. And by doing so, we get a frequency distribution of those different allele sizes or repeat lengths. And then we can do some fairly simple statistics to ask the question of whether all the individuals from one water column or one sample are genetically the same or different as that from another. These different allele frequency distributions identify gene pools, which can be used to identify genetically distinct populations. And how that variation is subdivided into distinct units tells us a lot about evolution. It tells us about selection. It tells us about migration, recombination, and the potential effects of genetic drift. So, as Elisa said, we have all this evolutionary theory we can draw upon, right? And so evolutionary theory tells us that populations are where it's at out in the field. We need to know what that distribution or that shape is of those populations. And we can start to make inferences about what's going on evolutionarily. So we are working with Thalassius aerochula. We wanted to first understand what's the global diversity of this organism. So we sampled 20 global locations. They're highlighted here in this map, isolated 50 single cells from each location at multiple time points. We extract the DNA, we PCR amplify the microsatellites. And what we found in the 449 individuals that we genotyped, we found 447 different genotypes. They're all different out there. We have almost as much diversity in this room. So that's a lot of raw material for contemporary evolution. We also know there are large population, large census sizes, right? Any of us that have done microscopy and do cell counts and say, okay, well, there's 1,000 cells per liter. That's a census size. We also know there's a large, it's called effective population size, a large genetic size because of all that diversity. And what that tells us, really importantly, is that genetic drift is not going to be as important as some of those other mechanisms because we're probably dealing with very large populations that aren't as subject to the vagaries of random movement in their allele frequencies. Okay, so we know there's high diversity. We might ask the next question, which is how is it subdivided? So we can quantify the divergence between water samples, all the individuals within water samples. We test for that. We measure it using FST, which varies from 0 to 1. 0 being no divergence, 1 being complete divergence. And we test for isolation by distance. And we do this going back to our population genetics theory using some approaches developed by Wright in the 1930s, where we look at pairwise distance, so geographic distance between samples in kilometers, and then the amount of genetic divergence. There's isolation by distance. You get increasing divergence with distance. There's no isolation, you don't see that. This tells us a little bit about the ability for organisms to migrate from one place to another and tells us about gene flow. Okay, so I'll put it all out there at once. Here's the study results from the Atlantic Basin, the Pacific Basin, and all the samples globally. And what you can see is that there's no significant association with distance. Here's log distance on the x-axis and genetic distance, a normalized genetic distance on the y. There's no significant increase of genetic divergence with distance in the Atlantic, maybe a little bit in the Pacific. And while the line is significant, the R-squared value is very low for global, the whole globe. Oops, go back. So basically, there's no substantive isolation by distance on a global scale. What I want you to note, and we'll focus on this example here, is that we can go out to something like 10,000 kilometers. And this was distance, like shortest ocean distance, not the current distance, just shortest ocean distance. You can have samples that are very far apart, showing no genetic divergence, as well as very high genetic divergence. So distance isn't a good indicator of whether there's isolation. The same with samples collected in the same location repeatedly over time. No divergence to quite substantive divergence. So now we might continue to ask, how is that divergence structured on a map? Well, what we found were there were 6 genetically distinct populations, and some were sampled repeatedly. I'm not going to go into showing you all the data. I'm just going to map out those populations onto this global map so that you can see where they differ or how they differ. So here are an example of some of those locations. You can see 5 different populations here. Each population is represented by a different color. So South Africa, here on the East Coast, 2 samples very close together out on the West Coast. This is California off Scripps here, and then that's in the English Channel. So we see, interestingly enough, these 2 white populations, we see the same population in different ocean basins. Okay? So that tells us that this particular population has the ability to transport ocean basins. We also see the same population succession in different ocean basins. So when we go back and we resample those 2 locations that had the white population, we see that there's now a genetically different population there further on in the springtime. So we see some parallel succession of populations within the species. So the bottom line here is that there is population variation, but it's not static. We see changes in one location over time, and we see that these populations are not dispersal limited. Okay, so what do we learn here? Based on some really nice modeling of microbial evolution that Ferdy Hellweger and colleagues did a few years ago, and I recommend reading if you're interested in that sort of thing, we infer that these diatom populations are evolving more slowly than the connectivity of the surface ocean. If they were evolving more rapidly, we might expect to see biogeographies in different locations. But that's not what we see. And so based on some of this modeling work, the implication is that they're evolving more slowly. I'll tell you more about that in a minute. We also see that divergence is probably maintained by selection. We see that the populations are associated with both temperature and chlorophyll A, so both ecological and environmental selection. And what we know is that selection reduces gene flow between populations. So it's a way that these populations may be maintained over time. Now I want to give you a short tour through the literature to give you a few examples of what else might be going on out there. The first example is the Diatom Skeletonema marinoi, sampled from here off the coast of Denmark, and then a fjord in Denmark. And what they found is that they found very long-lived populations in this fjord in Denmark. By looking down into the sediment, they found that the same population was present over timescales of 100 years. That's a lot of time for evolution to take place. Outside, the waters outside of this fjord had a completely different population. Which brings up this question of whether fjords are a good birthing place for populations. We've seen the same sort of patterns now in 3 different diatoms and in different fjords, which is something that I won't be able to talk more about today. Now we can look at space and also taxa. So this is another global study from the Diatom Pseudonychia pungens. And they found different populations. Each color block indicates a different population from different locations. And they found the exact opposite results that we did. This is genetic similarity. So it's a flipping of that axis I told you about. So samples that are close in space are very similar. Far in space are different. So what they see were both distinct populations, but this increasing divergence with increasing distance. So perhaps in these pennate diatoms, there are barriers to dispersal, which I said is really different. So that begs the question, is there a pennate-centric signal? Are there different evolutionary potentials in different taxa? And that's something important for us to consider. What else do we know? So these colors represent perhaps something like these very, very distinct different populations. Most of these have been sampled along the coasts. we went out as part of the North Atlantic Bloom experiment to see what happens out in the middle of the ocean. And this is what we found. Each of these tiny little vertical lines represents the genetic composition of one diatom that we pulled out of the water. And you see these nice patches? Looks like this, except we've ordered these. All of these individuals are sampled from within this patch that we followed during the bloom. So we see a very different pattern where within one water mass we see this evidence of different populations. And we think perhaps this open ocean area might be an active mixing zone and a genetic repository of variation. How that plays out in terms of recombination is a story I don't have time to tell you. Okay, so in the next very few minutes, I'm going to tell you about diatoms and their friends. This is work that my student Olivia Ahern is doing. You know about the importance of bacteria to biogeochemical cycles, so I'm going to skip through that. We used these same populations that you're familiar with, and we looked at the intra-specific variation based on some teaser data that we had from a different diatom. We hypothesized there'd be a single core microbiome with shifts in composition between these populations. All right, so all diatoms, all Thalassiosauria rotula, single core microbiome. Maybe different populations would have different associates. When we isolate these rotula chains, we isolate them into, this is supposed to be microliters, about 200 microliters. We transfer. This dilution series means that we're transferring probably far less than one free-floating bacteria into the culture well. So we're probably just transferring those associates of the diatoms. We isolate we culture the isolates for 2 weeks, we extract their DNA, and then we sequence the bacteria to determine the composition. And I'll get right to the punchline, and that is that T. rotula has no core microbiome. In fact, no single OTU was present across all isolates. And the lowest taxonomic rank was at the order level. But many bacterial taxa were shared among isolates from a single water sample. And here I've narrowed this down just to show you one water sample out of that diversity. And you can see these are 10 randomly selected individuals, and they have a very similar interaction. We see that there are differences, and it's not just what was going on in the water column. These are 2 whole seawater samples shown on a principal components analysis, and these are the community compositions of the cells that were cultured from that water column. So they're not just reflecting what was in the water column. And then the last thing I want to tell you about is that this bacterial community composition actually maps directly onto the diatom populations. So remember, I showed you this picture before. It's the same picture for the bacterial populations. Note these 2 white populations here. If I look at the bacterial community composition for those, they also switch significantly, and they're also the same. So what this suggests is that there are very long-term interactions between bacteria and diatoms, and at that population level. Remember, that's where the evolution is happening, at populations. So it gives us this potential for coevolutionary dynamics between the two, and perhaps a significant impact on the function, survival, and biogeochemical cycling of these organisms. Okay, so I'm going to wrap up with answering some of those questions I started with. So the answer is that evolutionary potential is high. I haven't had a chance to tell you about this, but there are studies in other taxa that indicate the same. The mechanisms, what are those evolutionary mechanisms? We know that selection is happening, and it's influenced both by environment and ecology. We think genetic drift isn't so important because of the large populations. Some open questions are, what's the frequency of sex? It's really hard to measure that in the field. And then what are rates of mutation in the field? And one of the reasons that laboratory experiments are so important is they can give us a sense of what that mutation rate looks like. We know these populations are really persistent. Their lifetimes exceed the global connectivity of surface waters and perhaps longer than 100 years. And it's sufficient duration for these populations to acquire unique microbiomes. And in terms of that larger seascape question, we see that there are no barriers to dispersal in centrics and possibly, yes, in pennates. But remember, there's really only one study of each right now. So it's almost hard to pull out a trend. But if there are barriers to dispersal, if there aren't barriers to dispersal, that means there's a high probability of immigration, emigration, and niche occupation. So we can think about a changing environment and how those organisms will respond, as opposed to, let's say, this pennate diatom example, where you get a potential for local adaptation, but there's reduced immigration and gene flow, and perhaps the potential for extinction if the rates of environmental change exceed the ability of that population to evolve. And then finally, we have some indication that perhaps the open ocean is this genetic mixing zone that is perhaps yet another genetic repository. And with that, I want to just show this very long list of acknowledgments for the global sampling. If you look carefully, you will see some of your friends on that list. And we thank them heartily. We couldn't have done it without them. Thank you. I'm going to take like 1 or 2 questions, and then we'll take a break for. I'll wait till the. Tammy has a question. Tammy's bouncing up and down on her.
I'm so excited.
I got the mic. Tatiana, this may be a silly question, but I'm going to ask it anyway because I'm old and I don't mind anymore. In all of these different studies, do the different investigators define populations based on the same genetic criteria? In other words, I find myself wondering sometimes whether differences or similarities that we see are really underlying differences, or whether it just depends on how we've defined them. Because I know, okay, does that make sense?
Yeah, it's a great question. Well, so the good thing for us oceanographers who do this, is the field of population genetics is really quite mature. And so we can take the tools, the theory, the statistics from that field and apply them. And so there are some very standard ways to infer population divergence and determine whether they're significantly different populations. So I would say, yes, we all do it in a very similar way. There are some differences, and in fact, one paper where we were finding unexpected results, we actually did the population genetic analysis 3 completely different ways, and they all pointed to the same answer. So that's another way that we do that.
That's sort of why I was wondering, because I have seen it done different ways, and it always worries me.
Heidi.
Hey, Tatiana.
Who was on that list, by the way? Heidi was on that list of acknowledgments. Yes.
I'm interested in this idea about the North Atlantic gyre or open ocean as a mixing zone, and it went by kind of fast. Was the mixture of types that you saw in the open ocean actually the same as the types that you saw in the coastal waters distributed around the basin?
Yeah, so great question. It did go by quickly. We didn't know that was what we were going to find, and we worked with an organism that was Thalassiophyra gravida that we saw in that spring bloom. So we don't actually have coastal samples of that organism around the coast. That was an example of what we think is happening. The only actual sample we have is that open ocean sample. But when we look in the literature, we don't see that pattern of what look like distinct populations within one water mass. And in fact, it actually took us a while to identify that was the case, because we hadn't ordered it so nicely and said, oh, look, it's very clear. It was all mixed up, a jumble, and I was like, I don't know what's going on. I can't make sense of it. But it was only when we started to think about it in the context of mixing populations that it was. But yeah, it would be a really interesting follow-up for that particular species to better understand whether it's always like that or whether we see that just in the open ocean.